Hello and hi everyone. So today I'm going to talk about polymerase chain reaction, PCR. So PCR technique is just a process of copying the DNA. We amplify the mother of the DNA that we get, so we get a lot of increase in numbers of the DNA itself. So this technique is invented by this guy called POLYMERASE in 1987. So why do we copy DNA actually? Why do we make copies of it? Why we want to make an increase in number? So suppose that you have a blood smear from a crime scene that this DNA is from the tissue of the robber. So you don't want to kickstart analyzing with this much of DNA, because if something goes wrong, it's a complete proof of loss and the evidence is gone. So what we do is we amplify this DNA so it get a copy of this DNA itself. We make much more copies, from 2 to 4, and from 4 and 6, and from 6 to 8, and so on forth.
Others use of PCR include genetic study. You want to detect a mutation inside of the gene, and genome mapping, and also a forensic study. So PCR is an in vitro DNA replication, so you must perform an experiment in order to do PCR. So this major element needed for PCR is this 4 major element is needed, or otherwise you're not gonna start analyzing it. So you need DNA polymerase. There's a DNA polymerase. Usually we use it from an organism called Thermus aquaticus. So you must have your DNA template, your gene of interest, nucleotides, and primers. So let's kickstart with the denaturation step. The first step of PCR is the denaturation step. So you have a DNA, the gene of interest here. So let me get this into a 2-strand. This is your original strands. So you might want to denature it in a 94 degrees Celsius. So you perform at this temperature. So we'll get this strand. It was separated, and we use it from the Thermus aquaticus because it is a stable enzyme. It doesn't get denatured easily at high temperature, and the optimum temperature will be at 70 degrees Celsius. So next step is annealing. So how does the separated DNA is annealed? So this, we start off with a primer hybridization here. So this is 5' to 3', 5' to 3'. This is the forward primer, and this is a reverse primer. It will hybridize to the template strand, so it will start off the replication. So next is the extension step. Extension step would be a primer there. The DNA polymerase will bind and start off the replication process. Then you will get a double-stranded DNA from this template and this template. You will get this 4 template. This is the new template and this original template, and you will get more and more when you do PCR. This is the first cycle, the second cycle, and you want to repeat more and more to get more copies of the DNA. So this is just the very basic principle of PCR.